the big data that I think everyone's really excited, and in myeloma, we always talk in terms of targets. And right now, the target on everybody's mind is BCMA, B-cell maturation antigen, CD269. Everyone is chomping at the bit to get BCMA therapies into the clinic. We saw some data on 3 different types of approaches for targeting BCMA: CAR T therapies, bifunctional antibodies or BiTEs, bispecific T-cell engagers, as well as antibody-drug conjugates. So let's take the CARTs, because those are the fancy, sexy ones that everyone's really talking about. We saw data from Orva-cel, Ida-cel, and from Cartitude. These data are rather impressive. Looking at patients that have had 5, 6, 7 lines and beyond, achieving extremely high response rates, some response rates being upward of 100%, with many of these patients achieving MRD negativity. I think we still don't know what is the optimal target, the optimal dosing strategy, which one of these CAR Ts is going to win out. Also, the big question of whether or not auto versus allo CAR Ts are going to have an impact. But clearly, these modalities of therapies are not far away, with Ida-cel likely being improved within the next 6 to 9 months. Very anxious to get those therapies in the hands of our patients. However, ultimately, we do recognize that there is significant toxicity. And much in the same way that we look at newly diagnosed myeloma patients now as being transplant eligible or transplant ineligible, we kind of think about BCMA therapy the same way, that if you are quote unquote transplant eligible and we're going to give you a BCMA therapy, it should probably be a CAR T. If you're older or frailer or not quite transplant eligible, we should give you some other off-the-shelf product targeting BCMA. And here we have both antibody-drug conjugates and bifunctional antibodies. Well, some of the more interesting data coming out of the DREAM series of studies. These are the litany of studies looking at belantamab mafodotin, which is an anti-BCMA antibody-drug conjugate targeting, attached to MMAF, excuse me. They presented data from the DREAM-2 and the DREAM-6 trial, with a peek ahead at the DREAM-9 trial, which is a trial in progress. So DREAM-2 is kind of the doublet, just belantamab mafodotin, a single agent, belantamab mafodotin in advanced patients, having around a 31% response rate. Very interesting new option for patients, but obviously we look to combining these drugs that improve their efficacy. So looking at the DREAM-6 protocol, combining belantamab with bortezomib, seeing significantly high response rates, pretty deep and durable responses, and this is likely to be one of the early triplets. And I think a lot of drugs have gone this way, much as where selinexor was approved with the doublet, and now we see the Boston trial, selinexor and bortezomib reading out. That's going to be the next triplet there. Bortezomib and belantamab mafodotin is likely to be the next triplet there. And DREAM-9 is really a very interesting study, trying to push this drug up front, looking at RVD, kind of gold standard that we've seen at ASCO from the Endurance trial. But here we're going to look in DREAM-9 at using RVD plus belantamab mafodotin to see if we can deepen and improve even those induction regimens. So very exciting data from there. And I have a big interest in bifunctional antibodies. A lot of our patients may not be healthy enough for things like CAR Ts, or we may need something immediately for a rapid progression, and a CAR T may not make functional sense. So the data on teclistamab, the J&J BCMA-based antibody, was quite impressive. This data presented by Saad Usmani. And, I think we're all very impressed so far with a way to engage our own immune system, engage the T cells without the need to develop a whole CAR T platform. There is still the concern for cytokine release, CRS, with some of the bifunctional antibodies. So really trying to iron that all out, but really excited about the next wave of therapies with drugs like teclistamab and belantamab mafodotin as they work their way into phase 2 and phase 3 studies.